Life is a symphony of molecular processes, better known as metabolism. As discussed in previous videos, the molecules that facilitate metabolism and all activities and actions of a living organism are proteins. This is the opening video on the process of synthesis of these crucial biological molecules. It's important to remember that cells do not produce all the proteins randomly at all times. Instead, the cells determine which proteins are produced at what specific time. But how do cells control the order of production of proteins at a given time? The process of transcription is the first stage of protein synthesis. This is when the language of life, namely the nucleotide sequence of a gene, is transcribed to form the codon sequence of a messenger RNA. A crucial process that converts the genetic information stored in DNA into a format that cells can interpret and use to build proteins. Let's first take a closer look at genes. The DNA molecule codes for the genome of an organism, that is, the full list of all the genes of an organism. View DNA nucleotides as the alphabet of the language of life, and genes as paragraphs in the book represented by the genome. Each gene within DNA details the instructions for building a specific protein. But how do cells know which gene to express and when? This is where transcription factors come into play. Transcription factors determine the order of expression of these paragraphs, or genes that control the metabolism of the cells of the organism. They are specialized proteins that bind to specific DNA sequences and regulate the transcription of genes. They act as switches, turning genes on or off in response to cellular cues and environmental signals. These cues can include hormones, nutrients, or even stress factors. Some transcription factors promote the binding of the transcription machinery to DNA, thereby activating gene expression. Others block the transcription machinery, effectively silencing gene expression. The intricate interplay of transcription factors ensures that the right genes are expressed at the right time and in the right amounts. Within the vast expanse of DNA, promoter regions serve as signposts, marking the starting point of a gene. These short DNA sequences, typically located upstream of the gene they regulate, act as landing sites for the molecular machinery responsible for transcription. Think of the promoter as a launchpad for a rocket. Just as a rocket needs a specific launchpad to lift off, the transcription machinery requires a promoter to initiate transcription. The promoter contains specific DNA sequences that are recognized and bound by proteins called transcription factors. The binding of transcription factors to the promoter region helps to recruit and position the transcription complex, the molecular machine responsible for synthesizing a messenger RNA molecule from the gene. Without a promoter, the transcription machinery would not know where to bind to DNA or how to initiate the transcription process. The transcription complex is a remarkable molecular machine, a complex assembly of proteins and enzymes that work together to carry out the intricate process of transcription. At the heart of this complex lies RNA polymerase, the enzyme responsible for synthesizing RNA from a DNA template based on complementarity of nucleotides. Note that RNA molecules contain uracil in place of thymine. Think of RNA polymerase as a high-speed copy machine moving in the 3 to 5 direction. It uses one strand of DNA as a template to create a complementary RNA molecule, a process that involves unwinding the DNA double helix by helicase enzyme, which breaks the hydrogen bonds between the DNA complementary nucleotides. Complementary RNA nucleotides are added one by one to the DNA template strand. The transcription complex also includes a variety of other proteins that assist in the transcription process, including TFI-IA, IZIB, IID, IE, ER, and N2H. The roles of these proteins will be covered in detail in future videos. To summarize, some transcription factor proteins help to stabilize the transcription bubble, the region of DNA that is actively being transcribed. Still, others help to process the newly synthesized RNA molecule. We will look at the actual process of transcription in the next video. Thank you for watching. If you liked this video, do remember to press the like button, subscribe, and hit the notification bell to be alerted to future videos. Most importantly, stay curious.